welcome everybody to this conference, or not conference, this lecture, I should call it. This is the 5th annual Myles Hollander lecture. And I'm pleased to be here to help open and recognize our distinguished speaker, who I just had a chance to talk with a little bit. His name is Dr. Robert Kass, and he's from Carnegie Mellon University. And shortly, he'll be formally introduced by his former student, Elizabeth Slate. Let me say just a few words about Myles, my friend Myles, who's sitting back there. He has generously endowed this lectureship, and it's a successful one. We've had great speakers, and another one today. Myles had a great career here at FSU. He really did it all. He was a great statistician, award-winning statistician. In fact, I learned that he won the Godfrey Nurture Senior Scholar Award many years ago, which did something in my brain because Emmy Nurture, as a mathematician who was in my field, in fact, a pioneer, turns out Godfrey is her nephew. So there's a connection there, Myles, between you and me, abstract connection. Myles served as department chair for many years, and he had a lot of PhD students, 19 in all. His accomplishments, as I mentioned, led to lots of recognitions. He really helped put Florida State University statistics on the map, internationally, certainly here on campus, but internationally, and helped it be the great department that it is today. And it really is one of our top departments. Among his honors is the highest honor that you can actually hold internally here at FSU, and that's the Lawton Professorship, which he was named back in 1998. So it's really a great honor for me to help kick off this lecture for another year. And right now it's my pleasure to introduce Myles, who wants to say this.
I thank the audience, those attending in person and those looking in via Zoom. As an undergraduate at Carnegie Tech, I was happy to learn that one could have a stimulating career in statistics, both theoretical and applied statistics. At Carnegie, I was motivated by the statisticians Maurice and Ed Olds. Maurice was a mathematical statistician and a Bayesian. Ed specialized in quality control and industrial statistics. Then when I started graduate school at Stanford, my major professor, Lincoln Moses, sparked my love for nonparametric statistics and biostatistics. When I joined Florida State in 1965, it was a young department. It had been founded 6 years earlier by Ralph Bradley and already had 3 well-known professors in nonparametrics: Frank Wilcoxon, Richard Savage, and Ralph Bradley. Shortly after, Jairam Sethuraman and Frank Proshan arrived. And I enjoyed long, prolific research collaborations with Frank and Sethu. FSU also had excellent students who I was fortunate to work with: Ed Supina, Ramesh Korwar, Greg Campbell, Brett Presnell, many more. I endow this lecture series in appreciation of the field of statistics, Florida State University, and its statistics department. And I don't mean to boast, but this series has had a distinguished start with lecturers Nancy Reed, University of Toronto, Susan Murphy, Harvard University, Trevor Hasty, Stanford University, Adrian Raftery, University of Washington, and today Rob Kass of Carnegie Mellon University. Each of these distinguished lecturers come equipped with numerous honors and recognitions, including being members of the National Academy of Sciences. In a moment, my colleague Elizabeth Slate will say more about Rob. Elizabeth is an FSU Distinguished Research Professor and has many honors, including being a Fellow of the American Statistical Association, a Fellow of the Institute of Mathematical Statistics, and an elected member of the International Statistical Institute. At FSU, Elizabeth is the Duncan McLean and Pearl Levine Fairweather Professor. Elizabeth's talents, determination, and creativity have made this a tremendous lecture series. Elizabeth will now introduce today's lecturer, Rob Kass.
Thank you, Myles. It's my great pleasure to introduce the 2024 Myles Hollander Distinguished Lecturer, Dr. Rob Kass. Dr. Kass is the Maurice Falk University Professor of Statistics and Computational Neuroscience in the Department of Statistics and Data Science, the Machine Learning Department, and the Neuroscience Institute at Carnegie Mellon University. He earned a bachelor's in mathematics at Antioch College in 1975 and his doctorate in statistics from the University of Chicago in 1980. After postdoctoral work at Princeton University, he joined the Department of Statistics at Carnegie Mellon University in 1981. I know a thing or two about CMU, having studied there. It is an environment of rigorous scientific inquiry, one where Rob has thrived, building a distinguished and impactful career. At CMU, Rob has supervised over 20 PhD students, served as department chair for 9 years, and also served 3 years as interim director of the precursor to the CMU Neuroscience Institute. Rob's early research formed the basis for his co-authored book, Geometrical Foundations of Asymptotic Inference, and his subsequent work has been in Bayesian inference and the application of statistics to neuroscience. A unifying goal is understanding and improving how reasoning from data produces reliable scientific knowledge. He is a leader in the application of statistics to neuroscience and co-authored the book, Analysis of Neural Data. Rob has been influential in both statistics and neuroscience, having served on numerous advisory and award committees. He's held editorial roles for journals in both disciplines. He was the founding editor-in-chief of the leading journal in Bayesian statistics, the journal Bayesian Analysis. In addition to honors you've already heard about, Rob received the Outstanding Statistical Application Award from the American Statistical Association, and he received the Distinguished Achievement Award and lectureship from the Committee of Presidents of Statistical Societies. He is a fellow of the American Statistical Association, a fellow of the Institute of Mathematical Statistics, a fellow of the American Association for the Advancement of Science, and an elected member of the U.S. National Academy of Sciences. A note for Zoom attendees, please remain on mute during the talk and enter your questions into the Zoom chat window. We'll address those following Dr. Kass's talk. Rob's 2024 Myles Hollander Distinguished Lecture is entitled Reasoning from Data in Science. Please join me in welcoming Dr. Rob Kass.
Very nice. Thank you.
So this is a great event. I'm really honored to be here, and it actually feels sort of celebratory because it's really celebrating Myles and everything that your dean said about him. I will say I'm old enough to know that everything that your dean said about Myles is true. He obviously did have a big impact on the profession and on the department. So, you know, I've been a professor, as Elizabeth said, for 43 years. And I've adjusted very comfortably to almost always being the oldest person in the room. But it has its disadvantages. As a teenager, I was already an absent-minded professor, and at my advancing age, it can be downright dangerous. A few years ago, I was at an off-campus event, and I returned, walking back to my car, deep in thought, got in and had a moment of panic because the steering wheel was gone. And I thought, who would steal a steering wheel? And then I realized I'd gotten into the back seat. So I wish I were kidding. So increasingly distracted forgetfulness is one of the reasons I now script my talks. So the title I chose for this lecture, to which I've added a subtitle here, comes from an extended essay. An extended essay, a short book I'm working on called Reasoning from Data, the Foundation of Trustworthy Scientific Knowledge. I've been completely engrossed in this effort, and I decided to share with you some of my thoughts by identifying a few themes that run throughout the essay. In my abstract, I said these themes would be rigor, faith, and pragmatism. I will talk about pragmatism, but I added context. And then I seem to have had a senior moment when in my title, I changed pragmatism to progress. I mean, they do both start with P. So I will briefly explain what I mean by these four labels in the latter part of my talk. In the first portion, I'm going to discuss some results from a pair of papers based on data collected from multi-electrode arrays implanted in the brains of animals while they're doing some specific thing. But to begin, I need to give you some background. And some years ago, I came across a striking graphic that does a very nice job of illustrating something I think most of you already know, which is the brain is located in the head. And you'll also recall that neurons communicate through rapid electrical discharges known as action potentials. They take place over approximately 1 millisecond. This figure is a voltage trace recorded from the cell membrane, though all the voltage recordings in the data I'm talking about are recorded by electrodes that are outside the local neurons, as illustrated in this image, except that this is a single electrode at the tip of a very long thin metal shaft, whereas the data that we analyzed for what I'm showing today were collected from several types of multi-electrode arrays, 3 of which are pictured on the right. As is visible in the image at the extreme left, each electrode records voltage signals from many nearby neurons. Cutting-edge data and analytic methods are being developed by many people, especially in conjunction with the so-called Utah array and the neural pixels array. The latter especially has sparked a lot of interest because it packs a lot of electrodes densely into a small area. A typical neural pixels array has more than 1,000 electrodes available for recording. Several of our most recent papers and several still in progress have been based on data collected at the Allen Institute by Josh Siegel, who's managed to record from 6 neural pixels, each with more than 300 active electrodes, simultaneously in the brains of mice while they're shown a variety of visual images. Speaking as someone who's been working on analysis of neural recordings for roughly 25 years, it's an exciting time. We're finally, routinely getting the kind of data we started to envision more than 15 years ago. And for about the past 8 years, all trainees working with me have concentrated on identifying cross-population interactions, typically across different areas of the brain and their timing. Specifically, we've examined two different forms of data recorded from these electrodes: spike trains and local field potentials, LFPs, as I'll explain. This figure was created by former Carnegie Mellon student Ryan Kelly, who became my postdoc for 18 months before going to Google. Panel C shows the raw voltage recordings from all the electrodes on the left, along with a pair of displays on the right, based on a trace from one of the electrodes. The top right part of panel C shows a low-pass filtered version of the voltage. In statistics, we call it smooth, and it becomes the local field potential representing the aggregated neural activity of hundreds of neural synapses in the neighborhood of the electrode. The lower part of on the right of panel C shows a series of action potentials, or spikes, overlaid on top of each other. This results from a clustering procedure after high-pass filtering. One set of similar waveforms is in red and another is in blue, representing spikes from two neurons recorded on this electrode. Below them are the sequences of time points corresponding to these waveforms, which constitute the two neural spike trains. Here on the left is the image of the action potential I showed you earlier, and I want to point out that before the action potential occurs, you can see the voltage rising somewhat erratically toward a threshold at which the neuron fires. The voltage is often modeled as a random walk because the neuron receives inputs from both excitatory neurons, which increase the voltage, and inhibitory neurons, which decrease it. On the right, I'm showing you the variation of neural firing patterns, the spike trains, across 3 repetitions of the experiment called trials, across time, and across 4 neighboring neurons. It comes from a review that I organized, a large international effort aimed at summarizing concisely both mathematical and statistical modeling approaches. Neurophysiologists do their very best to make sure the experimental trial replications are as similar to each other as possible and independent. Like the bulk of the literature, at least until very recently, we have focused our attention on these repeated trial experiments. The main interest here is in the frequency with which neurons spike, in other words, the neuron firing rates. For example, this particular neurons fire rapidly in response to specific visual stimuli, as shown here. There are simple ways to reduce the variability. We can average across trials to produce a peristimulus time histogram, or PSTH, which will come up again in a few minutes. Or we can average across time, which creates spike counts. And again, this is over now a fairly substantial time window relative to the timing of these events. You can see at the bottom, several hundred milliseconds typically for a count. This is the most common summary that's analyzed in the literature. So I want to mention now some results on spike count correlation, often called noise correlation. Correlation across trials, it's across repeated trials, among spike counts from pairs of neurons across relatively long windows of time, as I just showed a second. Each data point in these histograms is a correlation value for a particular pair of neurons. The histogram on the left is based on 42 pairs of neurons, and the histogram on the right is based on 193 pairs of neurons. So you'll notice that while there's a lot of variation, there is on average a small positive correlation. This is a widely observed phenomenon, and its relevance to the functioning of circuits in the brain has been widely discussed, beginning especially with the 1998 paper by Mike Shadlin and Bill Newsom. They looked at other data to pick the Pearson correlation value of 0.12 as a typical value in their very influential article, which I use as one of the first readings in a course on computational neuroscience that I developed and have taught almost every year since 2011, called Statistical Models of the Brain. These guys are both famous, and at this point, I happen to know Mike Shadlin pretty well. However, both the peristimulus time histogram and the spike count correlation histogram lose information that could be useful. The first about trial-to-trial variation, and the second about precise spike timing. And another point, the consequences of which I'll note in a moment, is that spike counts are subject to Poisson-like noise. The usual approach nearly every statistician would take is to figure out what we want to learn from the data and then formulate a statistical model as a crucial step toward achieving that goal. To emphasize what I'm calling the usual approach, let me just say that as much as I'm very proud of many of our papers, I don't consider any of the stuff we've done to have been especially clever. I've just followed my understanding of what statisticians are supposed to do. Statistically, spike trains are modeled as point processes, meaning events that occur randomly across time, and these produce Poisson-like noise. When we look carefully, we can see that spike trains do not follow Poisson processes. However, at least in the cerebral cortex of alert animals and often in other parts of the brain, the spike trains are pretty similar Poisson processes. Now we finally get to the first study. I want to discuss, done in collaboration with Josh Siegel at the Allen Institute, as I mentioned. I already showed you this figure illustrating the placement of 6 neural pixels in the mouse visual system. The lead author on this paper was my PhD student, Yu Chen, who graduated in 2021. He analyzed the spike trains from dozens of neurons recorded from each of 3 areas in response to drifting gratings as pictured in the figure, with 15 repetitions in each of 40 experimental conditions based on 8 orientations, clockwise, counterclockwise orientations, and 5 frequencies of the gratings, the frequency being determined by the width and number of the alternating black and white bars in the image, which sat in front of the mouse. Drifting gratings are well known to drive responses in visual cortex. Here, I'm going to show you a few results involving just 2 areas, V1 and LM. V1 is also called primary visual cortex. It feeds into higher visual areas such as the lateral medial area, LM. Panel A shows what amounts to 2 smooth population-based periostimulus time histograms fitted with non-parametric regression here, which are firing rate curves in each area, effectively averaging all the neurons in each area, all the neurons that are reported, across all the trials. The y-axis says spikes per second, but it's understood in this context to be spikes per second per neuron. We called the 2 dominant peaks population bursts and labeled them peak 1 and peak 2. The first peak, shortly after stimulus onset, presumably is dominated by feed-forward connections from the retina to the thalamus to V1 and then to higher visual areas. But peak 2, which is much later, should involve both feed-forward and feedback connections, including those from other brain areas. I'm going to focus on the timing of peak 2, specifically the time at which the peak 2 maximal firing rate occurs. I was interested in peak timing because when many neurons in a population start to fire rapidly, we can presume that information is transmitted. And from a statistical perspective, by definition, lots of spikes occur near the time of maximal firing rate. So the time itself should be relatively well determined statistically. In particular, I was hoping that with some effort at least, we could find much higher correlations than those small ones I showed you for spike counts, by instead examining the correlation of peak timing across 2 areas. The project actually started with a pair of undergraduates, a PhD student at MIT and Hannah Douglas, who's now a PhD student at Princeton. I asked Brian to create population PSDHs for each trial and to smooth them. So that's similar to what I've shown in panel A, except that the data for the plots in panel A use all the trials, while what Brian did was he created the analogous smooth histograms for each trial separately. Next, he found the time of the peak 2 maximum in both V1 and LM, and then he plotted the time of the maximum for V1 against the time of the maxima for LM. So the result is shown in panel B. Again, each dot represents the pair of population peak 2 times on a particular trial. To my dismay, there was no visible association. and it turned out no statistically significant correlation, even with a pretty substantial number of trials, 600 trials, 15 repetitions of 40 conditions. So I figured this is a job for a PhD student. Meanwhile, I asked Hannah to look at the responses of individual neurons in each area, and she documented multiple distinct kinds of responses. For example, some went quiet for a short period after the stimulus onset. The upshot was that the neural responses were clearly inhomogeneous. Yu Chen did quite a lot of work on this and was able to produce the plot shown in panel C, which we labeled denoised as opposed to the naive plot in panel B. There is now a remarkably high correlation, especially for anyone used to seeing relatively small spike count correlations of the type I showed earlier. He estimated the correlation to be 0.87 with a standard error of 0.025. The other striking thing about this relationship is that the time lag from the peak in V1 to the peak in LM is very consistent across trials at 9.6 milliseconds with a standard deviation across trials of only 2 milliseconds, even though the trial-to-trial variability in peak times themselves in V1 and LM is large, with standard deviations of about 30 milliseconds. This large variability is visible in the plot, where there's a large range of values along the y-axis for V1 and likewise the x-axis for LM. You can see that in the plot. As I mentioned, there's been lots of discussion in the literature about the sources of spike count correlation and what it might mean for efficient transmission of information. The interesting thing here is that in Yu Chen's statistical model, the only sources of trial-to-trial variation are at the population level, with the only trial varying parameters being the peak 1 time, the peak 2 time, and the average firing rate. Yet, after a referee asked us to look at spike count correlation histograms, which turned out to be a good idea, for individual neuron pairs, which we did within each of the 3 areas and across all pairs of areas, the model fit, shown in black for one example here, agreed quite closely with the spike count correlation histograms. In other words, population level variation in timing and overall firing rate can produce the kind of spike count correlation we see in data, even if the neurons are otherwise firing independently of each other. So that's to say, theorizing about the meaning of all this, I don't know. To me, like a bunch of neurons start to fire, that's the story. You don't need some advanced theory to try to explain why there's correlation. The model itself has a general form that's familiar to anyone who's worked with Bayesian methods. It includes multiple familiar components: model-based clustering, non-parametric curve fitting, latent variables, time warping, point processes. This model would be complicated for a statistical novice, but these days it's pretty straightforward for a statistics PhD student. It just takes some diligent work for a few months. Conceptually, it's straightforward. Each population is condition specific. So the orientation of the drifting grading, as the orientation of the drifting grading changes, the neurons chosen to define the population changes, because some neurons actually respond to certain orientations and some to others, so this is like we're picking out the ones that are firing a lot. And the population has a condition-specific template for the way the firing rate varies across time. That, again, it's important, it turned out to be important to let that depend on each condition. Both the composition of the population and the time-varying firing rate template are learned from the data. So, the take-home messages were, first, this approach is based on relatively homogeneous populations. By the way, I will mention only about maybe 15 to 20 neurons in each population, even though there's like, I don't know, close to 100 probably that are reported. But boy, we were doing it condition by condition. We're just pulling out the really good ones automatically with this automated method. Second, the 3 trial varying parameters, peak 1 time, peak 2 time, and the average firing rate are all features with high data content, so they're estimated well. Third, in contrast to analyzing spike counts accumulated over relatively long time windows, point processes provide millisecond precision. which was important for getting the result, especially about 9.6 millisecond lag. You have to have something that's got millisecond precision. And in addition, the use of this point process model automatically corrects for attenuation correlation. If you're not familiar with the attenuation of correlation terminology in statistics, it's about 100 years old. And it's easily explained by this figure. We first generated a batch of highly correlated data shown as green dots, and then added noise independently to each of the two coordinates to get the red dots, with the two gray lines indicating the noise added to a particular green dot to get the corresponding red dot. Clearly, the red dots have smaller correlation. This is very typical of spike-trained data due to their large Poisson-like noise. But it's easy to correct for it or denoise the correlation by modeling the noise, as we showed in a paper about 15 years ago. And again, this kind of point process model does the denoising automatically. The second example I'm going to discuss involves the problem of identifying coordinated activity of LFP oscillations, local field potentials, where we collaborated with the well-known MIT neurophysiologist Earl Miller and one of his postdocs. It's based on some excellent work by two former PhD students, Natalie Klein and Josue Oriana, that produced a body of theory and methods we call torus graphs for reasons that will soon become obvious. Here I'm showing you an illustration of LFP oscillations in some other data where the raw LFP is smooth, which is to say filtered within a narrow frequency band. It's very easy to create neural circuits that oscillate. It may surprise people who don't know about this, never heard of it, that neural circuits oscillate, but it's very easy to show why, in sort of at a conceptual level, why they should be oscillating often. If you just use the excitatory and inhibitory neurons, starting with just two neurons, one of each type, that are connected to each other in both directions, when an input drives the excitatory neuron, it starts to fire, which then drives the inhibitory neuron, and the inhibitory neuron stops the firing of the excitatory neuron. But then the inhibitory neuron also stops firing. So without the inhibition anymore, the input makes the excitatory neuron start to fire again, and the whole process repeats, resulting in up and down oscillatory behavior. So there have been many, many more realistic network models of this phenomenon, and it's been linked to important cognitive processes. Earl Miller has been interested in the idea that oscillations having frequency roughly around 10 hertz, 10 cycles per second, 16, I'm saying 10, it's the wrong one. That's a different one. 16 hertz, called beta oscillations, could be involved in communication across brain areas during learning and memory. Specifically, communication across the prefrontal cortex and the hippocampus, which are parts of the brain known to be active during learning and memory tasks. The starting point is to look for correlations across trials between the phases in two oscillations in two different parts of the brain. As each phase shifts from one trial to another, we can examine the differences of the phases for the two areas. And in Earl's data, it's easy to see a noisy but fairly consistent phase difference across repeated trials. Given a pair of bandpass filtered LFPs, a standard approach is to compute their phases at a particular time point in each of many trials and measure the coupling of the phases. Essentially, it's a form of angular or circular correlation across trials. In the last panel on the right, we pick a specific time during each trial at which to measure the phase. If the oscillatory phases of the two LFPs tend to move forward or backward across trials together, roughly speaking, we observe what's called phase coupling, which would indicate coordinated activity across the two brain areas from which the LFPs are recorded. A commonly used measure called phase locking value, PLV, that's what it's called. It is a commonly used measure. The circular diagrams on the right show how it can measure concentration of angles, in this case, the difference in phases across the two filtered LFPs. Under certain restricted conditions, which are commonly found to hold, at least approximately, for neural oscillations and their phase measurements, there's a strong sense in which PLV is analogous to Pearson correlation for angular that is circular random variables. Phase locking is closely related to coherence, which anybody in time series would be probably more familiar with, but it's sometimes preferred because it involves only the phase differences, whereas coherence depends on the amplitude of the oscillations as well. In a paper that we published last year, I think we gave a pretty comprehensive discussion of this and related matters in terms of the multivariate complex valued normal distribution. Earl and his postdoc Scott Brinkat recorded 24 LFPs across prefrontal cortex, PFC, and 3 regions of the hippocampus during a memory retrieval task. They looked at pairs of LFPs using a PLV. But I wanted to develop a multivariate generalization that would allow us to look at all 24 phases simultaneously. And importantly, we really wanted a method that would involve a probabilistic graphical model. To remind you, a probabilistic graphical model allows us to examine coupling that is unique to a given pair of recordings in the sense that it remains after conditioning on the other recordings, that is, statistically holding them fixed. In this diagram of 3 variables represented, if this diagram of 3 variables represented a probabilistic graphical model, we would say that there's a unique dependence between x1 and x3 and between x2 and x3, but not between x1 and x3. That latter dependence is removed by, I said x1 and x3, x2 and x3, but not between x1 and x2. The latter is removed by conditioning on x3. So the problem we set out to solve was how to formulate a multivariate version of phase locking value that produces probabilistic graphical models. A great clue for this started with a plot of pairwise data, that is, the phase measurements for 2 of the filtered LFPs. A data histogram is shown at the bottom right, with yellow indicating high concentration of counts, and the bigger plot on the left is based on data simulated to be similar to the real data, but it's a little cleaner. If you look at each variable separately, which you can do with this plot by just mentally projecting the data onto the x and y axes, they're pretty uniformly distributed, shown here as angles between minus pi and pi. But clearly, they're also highly correlated. However, there's this weird cluster of points up in the upper left corner, which turns out to be easy to explain. In the plot, x1 tends to be a little ahead of x2. The points are mostly below the main diagonal, which means that when x1 gets to its maximal value of pi, it has to hop back to minus pi. But if x1 is just a little past pi, x2 hasn't gotten to pi yet. So in rectangular coordinates, the value of x1 is way over to the left, while the value of x2 is up at the top, just below pi. That's why we see this weird plot. But it's highly unaesthetic, and it's the kind of thing that has always been deeply irritating. It's an indication there has to be a better mathematical formulation. Well, what do rectangular coordinates represent? They represent points in the product of the real line with itself. The domain of an angle is not the whole real line, it's a circle. The product of 2 circles is a torus, and this picture represents one of the angles by blue and the other by red. So if we make the bivariate plot again on a torus after smoothing, so the yellow indicates high concentration again, we don't have any aberrant values. One of the students made a nice video to illustrate this, where he placed a red dot to indicate corresponding points in the 2 representations. You can see the rectangular, the dot hops. Obviously in the torus, it just keeps going. Torus is the more elegant representation. And to get a graphical model for phase coupling analysis, it's statistically natural to develop an exponential family on the multidimensional torus, where we consider the full exponential family having all 2-way interactions. In computational neuroscience, this is also called a maximal entropy model. We defined torus graph to be any subfamily of the exponential family that has some subset of 2-way interactions. And because of this construction, a torus graph is automatically a probabilistic graphical model. It is the analog for random angles of the Gaussian graphical model for random variables on the real line. The rest involves a lot of details, which I'll spare you. A nice proof of concept comes from the student's analysis of the phases recorded from, oh, I said this already, sorry. Recorded from, there we go. The linear probe having 5 electrodes in hippocampus area CA3. Their torus graph reproduces the spatial neighborhood structure of the probe as unique pairwise dependence, unique pairwise dependence, whereas PLV, being a purely pairwise measure, cannot produce a probabilistic graphical model, and its graphical diagram could be misinterpreted as indicating unique pairwise dependence of every LFP phase and every other LFP phase. Instead, most of those edges shown at the bottom represent non-unique dependence. So the full data analytic graphical representations are shown here. Substantively, the main finding is that the apparent dependence of PFC is the gray ones. You can see this key down below. The V is for ventral, it's the ventral part of PFC. The apparent dependence of PFC on the dentate gyrus, DG, that's in blue, that's the diagonal from going from the gray to the blue on the left, in the PLV plot, can be explained by non-unique dependence through CA3 and the subiculate sub. So this is intuitive. So again, on the right, you don't see that diagonal going from the blue to the gray. And this is intuitive for a memory retrieval task because the dentate gyrus is an area associated with input to the hippocampus rather than output. Okay, those are my 2 examples. Now, I'm going to talk about my themes: rigor, context, faith, and progress. Let me begin with the easiest: progress. Statistics is a field of enormous progress. In my 50 years studying statistics, almost 50 years, the progress has been huge. And yet, even though there have been changes that could be considered revolutionary, as I've lived through them, they've felt gradual. I've witnessed the step-by-step building knowledge. In the book I'm working on, I use lots of interesting, often famous real-life analyses to tell the story, and they all illustrate advances. I would like to think that our torus graph paper was a fairly typical example in a couple of ways. First, a really interesting fact about the paper is that the mathematical part could have been done 30 years earlier, but the statistical and computational implementation relied on recent research and could not have been done even 5 years earlier, which shows how important the continual progress is to the field of statistics. Second, the methods discussed there may or may not turn out to be used a lot, but I'm confident we provided a clearer understanding of multivariate phase coupling because we had articulated an appropriate general framework, and we showed how many methods that had appeared in either the statistics or computational neuroscience literature were actually equivalent to special cases of torus graph methods under certain restrictive assumptions. For example, a method devised by physicists based on coupled oscillators yields a special case, valid when 1, the margins are uniform, and 2, there's no negative dependence. An absence of negative dependence corresponds to zeroing out what's called pseudo-covariance in the complex valued multivariate normal representation, as we discussed in that 2023 paper. But in any case, this is a typical situation in computational neuroscience where physicists look at a data analytic problem without leaning on the teachings of statistics. So, what is different about the way those with statistical training approach data analysis? Emory Brown and I discussed this repeatedly about 20 years ago, and in the introduction to our book, we wrote, many researchers have excellent quantitative skills and intuitions, and in most published work, statistical procedures appear to be used correctly. Yet, in examining these papers, we have been struck repeatedly by the absence of what we might call statistical thinking, or application of the statistical paradigm, and a resulting loss of opportunity to make full and effective use of the data. Over the course of a few years, I asked many statistical colleagues what they thought was the chief characteristic of the way statisticians approach problems. They used differing verbal descriptions, but there was a strong consensus, which I like to summarize with 2 core tenets. First, statistical models are used to express knowledge and uncertainty about a signal in the presence of noise by formal methods of reasoning from data. And second, statistical methods can be analyzed to determine how well they're likely to perform. The wording here is slightly modified from a paper Emery and I wrote in 2009, where we added, this is not the only thing statisticians do or teach, but it is the part that identifies the way they think. I'm going to quickly run through 4 comments. The first clear demonstration of these 2 tenets were in R.A. Fisher's 1922 paper. Our use of models, what we call models, allows them to be anywhere from highly restrictive, parametric, small numbers of parameters, to highly flexible, non-parametric, semi-parametric. When a model is used to produce a method, analysis of it, tenet 2 above, can be under a different model. This is how we check sensitivity and robustness. Finally, models, broadly understood, are central to the statistical paradigm. In fact, what we now call a statistical model is the most far-reaching idea in statistical theory. I've already said that models are central to the statistical paradigm, which is what distinguishes the way statisticians think. Historically, Steve Stigler has pointed to least squares, this is Laplace through Legendre 1805, as key to the development of the concept of a statistical model. He came to the conclusion that least squares emerged in part because these early mathematical scientists were essentially thinking observation equals truth plus error, in which I like to substitute signal for truth and noise for error, and then generalize observation combines signal with noise. This is a simple way to summarize what statistical models are about. Statisticians know that statistical models depend on parameters ranging from a few to infinitely many. Stigler also pointed out that Fisher was the first to use this terminology systematically, and it is key to Fisher's novel conception. Finally, part of what's going on when we invoke a statistical model is that sample quantities are distinguished from corresponding population quantities. I prefer to use theoretical instead of population. Fisher spoke of hypothetical infinite populations, but the word hypothetical got dropped, which I consider a profound and unfortunate change of meaning. I'll come back to that in a moment. Okay, with all these comments about the importance of statistical models in mind, we can return to my summary of the statistical paradigm, and I think we would agree that the statistical paradigm is the source of rigor, especially when you use tenet 2, the evaluate. Despite train and LFP analytic methods I discussed were built on extensive prior research about performance of related methods in the statistics literature, as well as our own performance studies, part of the stuff we published. Next, I want to make one further observation about models. A probability density function, a PDF, is defined for a fixed value of a parameter. This is often written as f of x given theta, with that vertical line. In case you're not aware, the use of the vertical line to denote conditional probability is due to the geophysicist Harold Jeffreys, who, among many other things, wrote a pioneering book in 1931 on what we now call Bayesian statistics. This notation for a PDF, f of x given theta, has been widely adopted in statistical contexts, having no commitment to Bayesian reasoning, even though Jeffreys, of course, was purposeful in using it for a PDF. However, interestingly, Jeffreys often wrote for emphasis f given both theta and a, where a stands for all the many somethings we are taking into account. We're essentially conditioning on them when we write down and use a model. And they're almost never specified, at least not fully specified. This is the source of context. Real-life statistical procedures take place in a particular context, which is largely implicit. In the last two examples I discussed, in those two examples, the context included not only a lot of statistical knowledge about a lot of scientific knowledge about the phenomenon that was under investigation, but also very careful experimental methods. I feel we don't stress enough the ways in which interpretations of statistical analyses always depend on scientific context. I've become aware that failure to recognize this reality is widespread. Many scientists seem to think or act as if statistics steps out of its applied setting to deliver some kind of ethereal rendering of judgment. This seems to be at the root of many problematic applications. I've now started stating one particular aspect of this phenomenon as starkly and as dramatically as possible. There does not exist a universal, context-free method of quantifying strength of evidence. Getting working scientists to understand this simple lesson from our field, I'm pretty sure, would have a huge impact. It might also render obsolete the largely irrelevant diversion of debating the merits of p-values. My use of faith comes from something I said in a 2000 article called Statistical Inference, the Big Picture, as I was interpreting this figure. Before I quickly run through it, let me emphasize that I said I was attempting to describe the dominant philosophy of contemporary statistics, at least among statisticians committed to scientific applications. So I was not claiming anything original, except that I was trying to explain this philosophy explicitly. The figure is crucial, and it displays just a few things. Most importantly, the separation of the theoretical world of models from what we take to be the real world of data. The figure also indicates that theory is different from, but connected with, data, and statistical models are in some ways like and connected with scientific models, while conclusions straddle the real and theoretical worlds because they're based on data but are most often about models or theories. Because random variables and thus statistical models are abstract mathematical creations, they live in a theoretical world that is fundamentally distinct from the real world of data. We can do our best to be careful and to check the extent to which our theoretical assumptions seem to be reasonable for a particular set of data. But not only can we never be certain the assumptions are correct, in fact, because the assumptions themselves live in that abstract theoretical world, there is no logical way they could be correct. The statement that a model is correct, if taken literally, has no meaning. In writing this article in 2011, I had several motivations: to downplay the importance of Bayesian versus frequentist arguments, to emphasize the centrality of models, as I've already done here, to suggest fundamental change in the way we teach statistics. In explaining the interpretation of confidence intervals, for example, we should explicitly use phrases such as, if these assumptions were true, then the probability that an interval would cover the true value would be 0.95. The use of the subjunctive here is essential, being forced on us by the gap between the theoretical and real worlds. I feel we should also replace population mean with theoretical mean, or at least explain that the population mean is a theoretical quantity. My final motivation for writing that paper was to correct what I considered to be a philosophical misrepresentation by the late eminent Berkeley statisticians David Friedman and Leo Breiman. Breiman's famous 2001 essay, Statistical Modeling the Two Cultures, was reprinted for its 20th anniversary, together with commentaries, including my own, in which I took issue with several things he said. The particular point related to my big picture was that although Although he rightly put modeling at the center of the statistical approach, Breiman followed Friedman by saying that models could be justified only when there was a chance mechanism to generate the relevant probability assumptions. In his essay, Breiman used the word mechanism 14 times. To explain what Breiman meant by mechanism, I want to draw an analogy with one of the greatest achievements in neuroscience, the mechanistic description of the way action potentials are generated according to the Hodgkin-Huxley model, most often depicted in cartoon form as a series of events involving first sodium channels through which sodium ions enter the neuron, and then potassium channels through which potassium ions exit. The more complete description involves a set of coupled differential equations, and its success is truly amazing. I like to say that among the Nobel Prize-winning research accomplishments, this is one of the really good ones. It not only explained the generation of the action potential in a particular type of neuron, it also explained many related phenomena. When Breiman and Friedman invoked chance mechanisms, they wanted to limit statistical models to cases in which they would function by providing explanatory mechanisms as the Hodgkin-Huxley model did. They thought the probability distributions in statistical models ought to be generated as in gambling devices or in statistical or quantum physics, which, if taken seriously, would limit models to a tiny fraction of domains in which statistics is used. When I said random variables live in the theoretical world that is fundamentally distinct from the real world of data and pictured those worlds as separated, I recognized that crossing that bridge between the two worlds might reasonably be called a leap of faith. There's no logical way we can bridge between them. But I added, it's precisely that leap of faith that makes statistical reasoning possible in the vast majority of applications. This is the source of faith we must have in reasoning from data. We work hard and we do the best job we can, and then we just hope it's good enough. The kind of faith I'm describing takes a special and important form in statistics, but it's generally invoked whenever scientific conclusions are drawn. And like other forms of faith, it has an emotional component. The Hodgkin-Huxley model is simple and powerful. It's been replicated and applied in hundreds of studies. It inspires profound feelings of awe in all those who come to appreciate it. And yet, over the past 65 years, as scientists have probed in greater detail, of course, it often needs to be modified. There are many different kinds of neurons with dozens of different ion channels, not just two, and a lot more is known about ion channels and the way action potentials are generated and propagate. Here is a really nice description of what constitutes a good mathematical model in neuroscience, though it could apply much more generally. What makes a model good? Clearly, it must be based on biological reality, but modeling necessarily involves an abstraction of that reality. It is important to appreciate that a more detailed model is not necessarily a better model. A simple model that allows us to think about a phenomenon more clearly is more powerful than a model with underlying assumptions and mechanisms that are obscured by complexity. The purpose of modeling is to illuminate, and the ultimate test of a model is not simply that it makes predictions that can be tested experimentally, but whether it leads to a better understanding. No matter how detailed, no model can capture all aspects of the phenomenon being studied. As theoretical neuroscientist Idan Segev said, borrowing from Picasso, from Picasso's description of art, modeling is the lie that reveals the truth. Picasso said art is the lie that reveals the truth. My broader interpretation is that the abstract world of symbols, including math, words, and paintings, can reveal important aspects of reality, the reality of scientific data or of lived experience. Furthermore, the most profound feelings we get from science or art come not as much from an understanding of specific data or specific behaviors of real people or specific details of scenes, but rather from our sense that a theory or a novel or a painting can explain so much. In science, building an abstract theoretical world and then applying its consequences to the inherently separate and different real world has been a very successful strategy. Why the strategy has been successful is a great human mystery, one that stirs deep feelings. Yet, of course, the many successes allow us to say the frameworks are valuable. More than that, regardless of our emotional reaction to certain mathematical formulations, the value of applying theory to data is the sole justification for the process. Because statistics forces us to face, on a daily basis, the challenge of navigating between the theoretical world and the real world. We have come to realize that the credo, modeling is the lie that reveals the truth, must be replaced by the famous dictum of George Box, all models are wrong, but some are useful. I have called this the quintessential statistical attitude. It's an important change of emphasis, and it led me to label the contemporary philosophy of statistics that I was articulating with my big picture, statistical pragmatism. So I've tried to explain some of the ways that reasoning from data, statistics, provides the foundation of trustworthy scientific knowledge. And it feels appropriate to close with a very nice quote from Jeffries in the introduction to his book, 1931. He said, the use of statistical reasoning, which had been a despised and generally avoided subject, becomes the most fundamental and general guiding principle of the whole science. Thank you all.
We now
told Elizabeth that a lot of my talks, they don't generate questions easily. But sometimes the stuff at the end, somebody says, well, do you really mean it, and then we start talking.
Hello. So I'm a physicist. Don't know much about what you had to say, but could you put one of the last say 109 on? Yeah, that will do.
This is, you want the picture?
So that picture at the top, I must confess, I don't understand how that line that goes from data to conclusions exists, because you have to go through some form of model or reasoning to get to some conclusions. So,
yes, sure.
How do you have that? How
sure,
do you explain
that's
that line?
perfectly fair point. I mean, it's a shortcut. I know that's a good point. You're using, there's no question you're using, theoretical stuff to get to the conclusions. But don't forget, there's an arrow. Actually, it is in here. See the statistical models goes to conclusions? It's being combined with data. That's supposed to be the answer to your question. It's actually there. It just, you don't see there's not like data and statistical models together give you conclusions, but that's what the two arrows on the two sides mean. Yeah.
a human observes data and the neural processes lead to conclusions without actually any modeling?
Well, if you're talking about, for example, an AI type system, which can do that these days, those are statistical models. That's just what they are. I mean, I'm not saying it has to be simple. And to be fair to what Bryman was talking about, he was mostly criticizing models he thought were too simple. But he's throwing the baby out with the dishwasher, with the bathwater. I mean, now we use complicated models all the time. And then, like I said, I'm counting non-parametric infinite dimensional models as models. They're models. Yeah.
Hi, Professor Kass. Hi, really wonderful talk. Okay, so I still have a confusion about one thing. Okay, unless you are fully Bayesian, okay, how will you go from, like, you have the statistical models, and from that you get the data. But whatever from the data and what you analyze the data, then you want to make any statement with some certainty about what's happening in real life or what happening to the statistical models. How can you take that step without being fully Bayesian?
Okay, so it's an interesting question in philosophy. Where did I do the confidence interval part? So the point that I was trying to make, actually I made it, where did I make it? I think it's in the paper. I forget, doesn't matter. The point that I was making about confidence intervals, if these assumptions were true, then the probability that the interval would cover the true value would be 0.95 subjunctive, right? That's the key, is it has to be subjunctive in that sense, right? You with me? For confidence. Exactly the same subjunctive has to appear in a Bayesian formulation. It's the same thing. If these modeling assumptions were true, if the prior were this, then we would have, posterior probability, blah, 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 blah. But you can't lose the subjunctive. The subjunctive has to be there. That's the point. And that's why I don't think the Bayesian frequencies are actually that far apart in terms of, especially what we teach students. Like, they're practically, they're two different ways of, I always think of it two different languages, but they can say the same things. In real life and real applied problems, I don't think of them as that different. It's just two different ways of getting to, hopefully, the conclusions you want. So I don't see, I see the big philosophical point is the subjunctive part, the split. And then you have two different, obviously, logical systems within the theoretical world. In the theoretical world, they're very different, sure. But the way they interact with the real world, that's not different. It's still just theory. You can do all the Bayesian stuff you want. You're still just doing theory. And, you apply it to data, sure. But you haven't escaped the theoretical part just because you happen to be Bayesian and it sounds good. And this is as someone who worked on Bayesian methods for a long time, by the way.
Thank you for your wonderful talk. Thank you for bringing statistics on manifolds in the subject. And especially a paper that I know very well, the 2020 Annals of Applied Statistics. The question is a little bit different. Like you pointed yourself, it's one thing the theory and the other part is the applied part. So in that paper, to my recollection, you had two rhesus monkeys that were...
We're talking about the torus graph paper?
Right. So how would you make an inference about a population? Or
so this comes up, there are many people who are bugged about this with
Right.
non-human primates. There's a lot of papers that only use one monkey. Sometimes two, sometimes three. We had an interesting, my one personal experience that I like to talk about is, which I guess we have time, I can give you the whole story about three monkey case. But actually had to review a paper that I didn't like, even though I know the guy's quite good, that was arguing against the whole idea of using, a single subject or a small number of subjects, exactly what you're just saying. I said, you're misunderstanding what the point is here. The point is, in these monkey experiments, monkeys are really different. There's huge subject-to-subject variability, but they're not different about, like, do they have primary visual cortex? And do they have PFC? And do these areas communicate with each other? Like, that's not different. That's like going to be the same in every single monkey you look at. So there's a lot of broad stroke stuff that's absolutely the same in every single monkey. And you're going to be trying to make inferences about that kind of stuff that's actually essentially perfectly replicated. It's not the details. The details are vastly different. And if you do care about those kinds of details that are different, then you got to look at a lot of them. No question. The one that I was, if you want to hear about replication, I have a whole story about replication. Anyway. See if there's other questions. If not, I can tell that story.
I'd like to bring in a question from the Zoom chat.
Sure.
The question is, according to your perspective, how does it fit in studies using EEG data, which in nature is more noisy than LFP data? How does the perspective you presented fit in the studies using EEG data?
Well, the perspective doesn't change with any kind of data. It's the maybe the torus graph analysis kind of thing. I've been asked about that before. Can I do that on this one? The problem is EEGs. I mean, they're really useful for certain things, but they're really noisy, and they're also. I mean, I don't like them as much because they're not that specific spatially. But could you do it? Yes, you could sometimes. The thing is, for that kind of analysis to be effective, you need really strong, visible oscillations. I mean, I don't think it's going to make a lot of sense unless you can kind of see it. Maybe you can't see it as much as in the example I showed, where it's like clearly there. But if you drew a spectrogram, a periodogram, Fourier transform to get the frequency plot, it better be visible there, that oscillation. If you can't see it pretty easily, then I don't like when I read people analyzing. There's all kinds of EEG analyses where all they do is they bandpass filter. They can't see, but you can't see it with your eyes in the actual, in the spectrogram or in the periodogram, whatever they're, So that makes me really nervous because I think the bandpass filter, and you bandpass filter narrow enough, it's automatically a sine wave, right? It's like, I don't like those analyses. So my answer is it depends. It depends on the specifics. If you have a real strong oscillation, I don't see why this method couldn't be applied, if that's the question. You want my replication story or should I skip it?
Okay.
Oh, go ahead. Yeah.
But we were discussing yesterday a little bit at dinner about the problem of that a lot of the statistic we use in neuroscience implies multiple repetition of the same trials, right? The same condition. But in reality, in real life, often our brain use information to make decision on a single trial. And novelty, it's one of the main informative things of an experience, right? If we experience the same things over and over and again, it's different, and now we suddenly experience something novel. So our brain in a single trial, in a single time, has to integrate this information to then evoke a behavior. So I want to get your opinion on where we will go from a type of statistical approach to then analyze instead of multiple.
So there are two different. So did you get the question on Zoom? Yeah. Okay. So there are two different parts of that. One we talked about last night. The second one we didn't. Last night we talked about there's a lot of people trying to do experiments where you have a much more, so to speak, naturalistic setting, and you don't have repeated trials in the same sense. They're not stereotypical, one just trying to make it the same all the time. And I can give you, for everybody else's benefit, I can say the same thing I said last night, which is I know what people are trying to do, which is to use regression-like approaches or sort of, there are behavioral events that happen. And the thing that's not the same as when you do repeated trials is that all the stuff leading up to that behavioral event isn't the same. So it's going to be kind of different necessarily. But, if you have enough information about the behavior from a video or something, hopefully along, like throw all kinds of machine learning stuff at it, maybe you can characterize a bunch of stuff pretty well and then use regression-like approaches to solve that problem. I think that's what people are hoping. I haven't seen anything yet that does that, but That doesn't mean it's not already been done. The novelty feels to me a little different, and I don't know, because I actually haven't thought about it. But it feels like novel experiences have to be put into a context that exists. Otherwise, we wouldn't call them novel. We wouldn't recognize them as novel. And so they actually have to have certain characteristics that are not wholly novel in a certain sense. Like novel odor, novel, I mean, there's things you can do, like if we were talking about olfaction, you probably can invent an odor that there's no way this animal has ever smelled before, I assume. But it's going to be, yes, it's going to be recognized as, okay, that's different than what's here, but still, it has to be that the animal's using that information that it already knows in order to do something. So I think that's clear. And then typically novel experiences, if they're deemed important, they get tagged. I always think of this as part of what emotions are for in humans is you're tagging an event. It's like, oh, that was really like embarrassing. Gets tagged. All the things I seem to remember from my childhood are embarrassing, So it's getting tagged. And so like salience, it's like it's going up. Those are things, the mechanisms that are going to get worked out and so forth. So I don't know. That's all I can say. I think it's all stuff you would probably agree with. And it doesn't seem that, yeah, it's mysterious in the sense that we don't know, but I feel like it's going to get worked out. You'll get especially something like olfaction where you can really, I don't know if you've already tried this, but where you can do something that's somewhat novel and yet, you'll figure out like, how's that kind of working, right?
I wonder if I could just squeeze in one more question.
Sure.
The denoising process you described, it concerns me that you say you just sort of extracted the very good behaving data, if I paraphrase it a little.
Yeah.
And I wonder if you see potential for complicating the model to handle heterogeneity a little better, to maybe be able to extract some interesting signal from that stuff you had discarded.
In that particular case, not so much, because I think I think what's more likely going on. So there's a couple things. First of all, neurons respond to different orientations. This is like really well established. So a lot of what you're throwing away are the neurons that are responding to a different orientation than the one that you're showing on that particular trial. You don't want those because they're just contributing noise, actually. They're literally spiking at kind of weird times that don't have anything to do with the stimulus. You don't want those. Then there's this other heterogeneity that I alluded to that the undergraduate found, some neurons actually go quiet. Well, they're doing something, but this particular visual stimulus is probably not going to pick it up. So that's an experimental issue. So as it happens, this is just one of a whole bunch of stimuli. There's one student who came along and wants a project, and I'm like, what? And it's like, he's just kind of a random student. He's not going to be a PhD student from me. And I said, well, you can look at there's like, I hope like all these other stimuli that, why don't you try, doing the same analysis over again on some other, so maybe you discover some very different neurons that are doing some other things. But I think the typical thing in, like you take a primary visual cortex neuron, which is probably one of the most heavily studied neurons ever in any system, they're doing a lot of different things. And this task happened to have picked up one of the kinds of things they're doing. And so it's really, I would say, more of an experimental question than a statistical. And if you have a good experiment, then I would be trying to tease apart the different kinds of stimuli that then, you'd build it would be a more complicated model, but it wouldn't be so much that you're learning more about that peak timing thing from other neurons. I think that's unlikely. I think the only way you can do that is by getting more of those neurons, because there probably are more than just the 17 we happen to get. It's just that 17 was enough to do a lot. So is that a good enough answer?
Much. And at that, I want to take just a minute and thank everybody who helped facilitate this event. Firstly, Myles Hollander, thank you very much for making this possible. I want to thank Pam McGee, our business manager, and the whole Department of Statistics staff. I also want to thank Heather Athey. She's the Director of Media, Communications, and Marketing in our College of Arts and Sciences, and her team for making this possible. And I would like to present to Rob.
Oh. That's swag. Oh, how nice. Beautiful. That's so great. It's really nice. Thank you. Cool. There's someone taking a picture. Hold on. Nice. Beautiful. Very cool.